Well, welcome to Immunology for Physicians or Immunology for BPTs. I'm David Fulcher. This is the first Chapter 0 lecture in my course in Clinical Immunobiology. I've made it freely available. The idea of this lecture is to give you a general thumb sketch of the basics of the immunobiology that are more or less an entry level for the course. We cover a bit of stuff that we will go over and over in the course. So if you feel a little bit out of your depth in this lecture, or whether you don't know some of the aspects that we're talking about, don't worry, we will go through it all in detail. But this is just to give you a quick roadmap of the immune system in all its glory as a prelude to the more detailed material in the course. So this talk is in 3 parts. It refers to the Primer of Clinical Immunobiology, which I've called Chapter 0, to emphasize the fundamental nature of some of the things which I would think that most people who've studied medicine or doing biology would be on top of. But just to make sure that we don't sort of assume too much, we'll take things from a fairly basic level before we launch into the nitty-gritty of the course proper. So, immune system designed to protect ourselves from invasion by bugs. Bugs come into the system, it goes into the lymphoid tissue, we make a response. We're going to flesh this system out, obviously, in quite some detail in the course, but that's basically what we're talking about. And this had its roots in the initial discovery by Edward Jenner that if you infected people with cowpox or vaccinia causing cowpox, they had a relatively mild disease, but they were then protected from smallpox, which would otherwise have been a severe disease. So vaccinia gave rise to the word vaccination, and 200 years after this discovery, smallpox has essentially been eradicated. So this was a monumental event. And with our most recent infection, the same sorts of principles apply, with this fascinating graph showing significant protection from COVID-19 from vaccination compared to those who did not get vaccinated. So these are the principles that we are talking about, things that we do that the body does to protect itself from getting severe and nasty infections. Now, this starts with the differentiation between commensals and pathogens. So commensals are microorganisms that make up the microbiome. They live in symbiosis with the host. They don't penetrate the mucosal layer. They don't do damage. We more or less live in harmony. But this is distinct from microbes, which are pathogens. Pathogens coming from pathos, generating, makes us miserable. I learnt that by watching The Chase, so always pick up stuff looking around. Misery causing, so these are things like nasty viruses, bacteria, fungi, parasites, helminths, which do penetrate the mucosal layer, inducing damage to the epithelial cells and indeed spreading to other tissues. So to protect ourselves from pathogens, we have 2 layers of immune defense, which have their own respective sciences, which are fascinating in themselves. Traditionally, we learnt about the adaptive immune response or the specific immune response involving B or T cells, but more recently we've come to recognize the fundamental importance of cells of the innate immune system. So basically what happens is that we have this intrinsic series of defense which we share with other organisms all the way down to bacteria themselves, where a foreign pathogen or foreign invader is detected by specific receptors which then fire off to generate a rapid response. In complex organisms like ourselves, these bugs are recognized by the innate immune system, which brings about a rapid response which gains general control, but is not adequate in itself to protect the organism completely, but does send signals to the cells of the specific or adaptive immune system, which then generates a delayed response which is ultimately successful. So we're going to talk about those two arms separately, starting with the innate immune system. So innate immune defense is present in just about all organisms, either by themselves, like slugs and plants and even bacteria themselves, or linked to higher tiers of defense, such as in complex organisms such as ourselves. And this defense is to recognize intrinsically foreign motifs, to respond rapidly, to hold the fort, whilst the adaptive or specific immune responses can be generated. This innate system recognizes foreign motifs in the pathogen via innate receptors. These receptors are encoded in germline DNA, and this DNA is passed on to progeny in order to provide some basic protection intrinsic to that organism itself. Things like what we call antimicrobial peptides, which we'll come to shortly, complement you may know about. Some of the cells of the innate immune system, macrophages, neutrophils, which can just ingest foreign things and break them down and prevent them doing any damage. Once the innate immune system is activated, it can respond by releasing more antimicrobial peptides or other inflammatory antiviral mediators or cytokines, which we'll come back to. And once it's done its job, it also sends signals to the adaptive immune system in order to beef it up and make a definitive response. So the adaptive immune response is different. The innate immune response has a limited number of receptors of about 100 by which it can recognize foreign, but the adaptive immune system can recognize orders of magnitude higher numbers of specific motifs. So this system is obviously more successful than the innate. That's why we need it to protect an organism as complex as ourselves. And sort of unlike innate, memory is a factor. So once the immune response has been made by this specific immune system, the next time the bug tries to get in, the response will be that much more rapid, and indeed there will be things like antibodies preventing a secondary infection in some circumstances. So when we're talking about the adaptive or specific immune system, we're talking about antibodies, namely specific proteins that bind structures on pathogens. And we're talking about the cells that make antibodies, namely the B cells as well as the T cells. And we call these the antigen-specific lymphocytes because they are directed specifically at antigen. The abbreviation for antigen, AG, get used to it. You have to use it all the time in immunology, of course. So an antigen is any substance that can be recognized by the adaptive immune system and generate a response. Now, the adaptive response takes time. It requires clonal proliferation, which we'll talk about, a fundamental of immunology, and that takes a few days, and that's why we rely on the innate immune system to provide that initial protection whilst the adaptive immune system is beefing itself up. Now, we don't pass this protection on to our children, okay? If we've been vaccinated against measles, our children will not be born immune to measles. We'll need to repeat the vaccination with every generation. Unlike the innate immune system, which has a smaller number of receptors, which are all encoded in DNA, which is passed on to the progeny. All right, now the first thing that protects us, it's sort of innate-like, if you like, is the integument, basically the balloon that protects us from the outside world. It's not very interesting immunologically, but we can't ignore it. Basically, we have tight junctions on epithelial cells, mucus on mucosal lining. We have antibacterial enzymes along the surfaces, such as in tears, saliva. We have histatins, chemical barriers, acidic stomach, digestive enzymes. All of these things would be pathogen often would have to try to get through in order to reproduce itself, or in other words, cause an infection. Surfactant proteins also have a protective role and can mediate opsonization, which we'll come to very shortly. The immune cells that we will deal with throughout the course are hematopoietic cells derived from pluripotent stem cells in the marrow, and this applies to innate and adaptive immune systems. In general, these are generated throughout life and renewed throughout life, and they belong to the myeloid and the lymphoid lineages. Basically, cells are made in the marrow, move to the circulation throughout life, and then recirculate. And lymphocytes, of course, have their own system of recirculation called the lymphatic system, which we'll come to at the end of this lecture. Now, these are the innate immune cells, which I want to talk about one at a time, just one slide per innate immune system, innate immune cell, just to remind you of what all these things are doing and how they came about. And by and large, we're talking about these guys down the myeloid lineage, and these T and B cells are part of the adaptive, and the other innate cells are the NK and the ILC. So leave the B and Ts for the moment. We'll talk about all of the rest of these sequentially. So first of all, macrophage, big eater. Macro, big, phage, eater. These are long-lived cells generated during embryonic development. They are tissue-resident, self-renewing. They're also renewed from newly generated monocytes, and their job is to ingest pathogens that have crossed that epithelial barrier that we spoke about a moment ago. Now, they can ingest these pathogens in a number of ways, which I've outlined in this graphic here. They can do it non-specifically. In other words, they just munch away at whatever happens to be in the environment in which they find themselves, and if they don't like it, they spit it out. If they do like it, they break it down. But that's pretty inefficient, and so that we have ways of making that ingestion far more efficient. So if these surface receptors, for example, on a macrophage are engaged, they will specifically phagocytose whatever happens to be binding to these receptors. So we have innate receptors, which I'll come to shortly. We have some receptors for the antimicrobial peptides. We have receptors for complement and receptors for antibody. And so if a pathogen is coated with some of these substances, it makes it far more easy for the macrophage to ingest it. When the macrophage does ingest it, it breaks it down in its phagolysosome, which contains degradative enzymes and proteins to break down the substances. It's acidified, which makes it easier to break things down, and it generates toxic oxygen metabolites, which act to kill whatever pathogen happens to be in the phagolysosome. At the same time, if these receptors are engaged, it induces the macrophage to release mediators to cause local inflammation. And local inflammation is important. It's like signaling to the cavalry to come and get going. Something's going on here. Let's release these signals to get more cells into that area to fight whatever pathogen is trying it on. I also will just mention in passing that in this phagolysosome, the pathogen is broken down into small peptides, and these become relevant when we talk about how T cells are activated. So here's an example of a macrophage here ingesting a mycobacterium. It's doing so by a toll-like receptor, which we'll also come to shortly. Okay, that's macrophages. Neutrophils. Neutrophils are also phagocytes. They're generated in the marrow continuously. They migrate to blood. They only last a day or so, so they're rapidly renewed, and they move into tissues in response to any of these inflammatory mediators that happen to be being released by things like macrophages or other mediators of acute inflammation. So they move into the tissues, they ingest pathogens similarly to the way that macrophages did, and when they ingest them, these the phagosomes fuse with the prominent granules in a neutrophil which contain degradative enzymes and other proteins such as myeloperoxidase and proteinase, which break down that pathogen and kill it. At the same time, these cells, like the macrophage, take on oxygen. They generate this oxidative burst, which acts to generate superoxide and other oxygen radicals within the phagolysosome, which then acts to kill the pathogen. And that's an important part of neutrophil biology, which we'll come to when we talk about immunodeficiency diseases. Eosinophils. Eosinophil, they stain with eosin, hence the name. And basically the claim to fame for eosinophils, they do a fair bit, but the reason why we think they're there is because they are good at trying to kill a pathogen that's too big to get in a macrophage. So things like worms and helminths, parasites that you can't just get inside a cell. The eosinophil sort of fights back by trying to dump all these granules onto the pathogen and hopefully bring about its demise. So it's designed to kill these big pathogens extracellularly, a role that doesn't seem that effective, to be honest, and we mostly know them for their role in allergic disease. Similarly, mast cells and basophils are well known for their role in allergic disease, but of course that's not why that we have them. They leave their marrow in a mature form, they mature in the tissues, the gut, lung, skin, and when activated they release their resting granules. So they're called mast because mast, I think in German, means well-fed or fattened because they're so full of these blasted granules. So all these granules, once the cell is activated, they are released and become degranulated and release all the mediators that happen to be present in these granules, as well as make new mediators which mediate inflammation. So that's what they do. We mostly know them for their bad role in allergic disease, but they probably have a physiological role. It sounds likely that we'd have a cell that does something useful. Bit hard these days to look back through the history of biology and work out exactly why we have them. And a few things have been postulated, but basically, that's what they do. They may have a role in helminth protection, similar to eosinophils, but given our Western society, helminths is not such a big factor for us in many places of the world, and so their role is a little bit unclear. Basophils have a similar function to mast cells, except that they are mostly circulating and they migrate into tissues when needed, whereas mast cells sit in the tissues all the time. But basically their function overlaps quite significantly. All right, dendritic cells are important. Now we'll talk about 3 types of dendritic cells in this course, so excuse me, just be aware that not everything that says DC is the same. So I should call these classical dendritic cells, but sometimes you just get used to just calling them dendritic cells. The other 2 are follicular dendritic cells and plasmacytoid dendritic cells. They're all called by their prefix. If you see dendritic cells, we're talking about classical dendritic cells in most cases. But the claim to fame of dendritic cells is they're really important in activating the adaptive immune response. So they are activated by innate signals, but they mediate adaptive immune responses. Anyway, coming back to their biology, immature dendritic cells are released from the marrow, they migrate to the tissues, they lurk in the tissues waiting for a foreign pathogen to be recognized by their pathogen recognition receptors on their surface. They have a big surface area for this purpose, and when these innate receptors capture pathogen, they do 2 things. They hold it on their surface, and they also ingest it to break it down into peptides. The antigen on the surface is retained to help activate B cells, and the processed, broken down peptides, which occur in similar ways to macrophages, are then represented to activate T cells. So antigen-specific lymphocytes are activated by dendritic cells, which is their most important function. Now, we'll talk about, of course, those processes in this lecture and through the course. Okay, our final and perhaps most recently delineated innate cell are the innate lymphoid cells, or the ILCs. Now, we've known about cytotoxic innate lymphoid cells for many years, and we call those natural killer cells. So the natural killer cells are a significant proportion of our circulating lymphocytes, maybe 10-20%. They look, as down the microscope, they look like large granular lymphocytes, and they respond to a number of signals from virally infected or tumour cells, and they respond by killing their target, hence their name. On the other hand, the more recently discovered arm of the ILCs are the helper ILCs, as distinct to the cytotoxic ILCs, and these localize around surfaces. They also recognize danger signals released by those surfaces. A large number of possible signal molecules can trigger them, and basically they release cytokines, which then can govern local and distal immune responses. So more about ILCs later, but they are an integral part of also guiding the adaptive immune response following danger signals that come from pathogen encounter. So one of the corollaries of this is that the innate immune system engagement is fundamentally important for our immune responses. If we inject you, for example, suppose you want to make a hepatitis X vaccine, you can't just get the protein and inject it into someone, because that's a very poor immunogen. What you have to do is take that protein and make it look like a pathogen by mixing it with microbial structures, which are often called adjuvants, things that will engage the innate immune system strongly and beef up the immune response. So many of the new vaccines will contain structures called adjuvants, which will engage the innate immune response and increase the overall immune activation. And so basically we need to make it look foreign, and by foreign in an immunological sense means the ability to engage innate receptors. Well, here is a section on innate cell receptors that we've alluded to several times already. So there are about 100 receptors encoded in DNA passed on to progeny, and we call these PRRs, or pattern recognition receptors, for obvious reasons. You have a pattern on a bacterium, and we call these pathogen-associated molecular patterns, or PAMPs, and that pattern is recognized by pattern recognition receptors, be they soluble, be they membrane-bound, be they intracytoplasmic. Basically, we have receptors in all sites to cover all possibilities for the way that a pathogen may try to invade. So these are the types in a nutshell of pattern recognition receptors that the innate immune system uses to recognize foreign. So we have soluble particles, the soluble proteins such as the antimicrobial peptides and the complement components. And these can block the binding of the pathogen. They can also kill the pathogen, and they can also facilitate opsonization. Now, opsonization we will come back to all over the place in this course, so we might as well cover it now. It means the coating of a pathogen with molecules which facilitate its uptake by things like macrophages and neutrophils. So if you have a straight organism, I've told you that the macrophage will take that up a little bit, but if it's coated with antibody or complement or antimicrobial peptides, we say it's been opsonized, and that facilitates its subsequent ingestion and destruction. Okay, so that's what some soluble pattern recognition receptors will do. We also have membrane-bound receptors, and these fall into families such as the C-type lectin receptors and the toll-like receptors. And these can stick or bind the pathogen. Some of them can signal. Toll-like receptors almost always signal, and this provides a strong activating signal for that particular cell. Toll-like receptors can also be within vesicles, and these often recognize breakdown products of the pathogen and then send that activating signal. So it can not only be on the surface, but it can also be in the phagosomes. And we also have a series of receptors in the cytoplasm to detect foreign structures from a pathogen which has made it into the cytoplasm. Things like nod-like receptors, rig-like receptors, aim-like receptors are all very potent cellular activators. And one of the most important innate molecules is complement, and so much so that we spend 2 lectures talking about complement. So complement is a series of proteins activated sequentially in a cascade where each component is able to cleave many copies of the next component, and by cleaving those components it actually activates them to cleave downstream molecules within this cascade. So a small initiating event will therefore have a large distal effector event. Now we have 3 ways of activating complement through 3 different pathways, which we'll need to go through in some detail. But just to summarize it now, we have the lectin pathway. Now the lectin pathway is set off by mannose binding lectin, MBL, or phycolins, and these can recognize lots of different motifs, which we'll talk about. So a lot of pathogens can just bind MBL and set off the lectin pathway. The classical complement pathway requires mostly the role of antibodies, not exclusively, because CRP and C1q can act as pattern recognition receptors in themselves. More about that later. But suffice it to say, mostly the classical pathway requires antibody, either IgM or IgG, to set it off. And we have a third pathway called the alternate pathway, which is just set off by a surface that is foreign. And more about how that works later. The central step of all those pathways is to convert the third component of complement, C3, to split it into C3b and C3a. And C3b is a very strong opsonin, so it will coat its target and make it easier to be ingested by a macrophage. And it can also set off C5, 6, 7, 8, and 9 to make a membrane attack complex, which essentially drills a hole in the pathogen membrane and allows solutes and stuff to flow in, basically kills it. And the final thing that I want you to note at this stage is that during that complement activation, inflammatory mediators called anaflatoxins are released, C3a, C5a, which can mediate inflammation. So the function of complement in a nutshell: opsonization, you're all over that now, right? Killing its target by a membrane attack complex and releasing inflammatory mediators called anaflatoxins. Now, part of activation of innate immune cells is that they release other mediators, as I've outlined, and these are often vasoactive mediators. In other words, they act on blood vessels, which opens up those blood vessels, allows increased blood flow, and then the delivery of fluid into that area, as well as cells into that area. And the fluid, remember, will contain antibodies, complement, antimicrobial peptides, all sorts of things which will deal with the insult from the pathogen trying to invade. So vasoactive mediators, we recognise PAF is a very strong one. Doesn't feature a lot in clinical medicine, but it seems to be important. Histamine, leukotrienes and prostaglandins all do, and you will recognise those chemicals. So the other reason to increase blood flow into an area of inflammation is that it also increases lymphatic flow, and that flushes any pathogen down the lymphatics. They get trapped in lymph nodes where antigen-specific lymphocytes reside and can initiate a specific immune response. Now, vasoactive mediators do or act on the endothelium to activate it, and that, in other words, it makes it become stickier to immune cells and allows them to migrate into the tissues. And this includes neutrophils. Remember, neutrophils are generated throughout life and look for these activation of the endothelium, and that tells them where to go. Macrophages do the same thing. These come in, degrade, ingest, break down, and clear out any debris and then promote healing. The endothelium also can be activated by chemokines and other chemoattractants which also allow cells to come in to that area. More about those substances very shortly. And we know the cardinal manifestations of inflammation: swelling, redness, heat, and pain. So this is the activation of the endothelium. In a nutshell, endothelial cells are acted on by the anaphylotoxins, by chemoattractants, by various chemokines, which I'll talk about shortly, activating the endothelium and permitting the egress of fluid, delivering important molecules to deal with the local infection and flush them back through the lymphatics, but also bringing in cells of the of the immune system, innate cells, as well as specific lymphocytes, to come and deal with it and generate a specific immune response. Now, you'll hear a lot when you hear an immunologist get up to talk about cytokines and chemokines, and it's not that complicated. Cytokines are small molecular weight proteins, 100 or so. If you hear the words interleukin or interferon, you're dealing with a cytokine, but there's lots of other cytokines which don't start with that. And they are basically designed to act locally, but some of them will act systemically. In other words, they will act on either the cells within the area and beef those up, or they can be released throughout the body and turn on an immune response. Interferons, tumor necrosis factor, interleukin-6 all fall into that category of acting more systemically to basically put the body on alert, sometimes shut it down while a pathogen is being dealt with. These sort of, in many ways, coordinate the immune response in doing so. Now, chemokines are a subset of cytokines with the specific role of attracting particular types of leukocytes into the area of invasion. And we'll talk about the chemokine system in some detail, but there's a stack of chemokines and a stack of receptors, and the different combination of chemokines and receptors will govern the nature of the inflammatory infiltrate, which will be drawn into a site of pathogen invasion. All right, now, enough about the innate immune system. Let's move on to the adaptive immune system. The idea of the adaptive immune system is that they have surface receptors that are directed at a specific structure. These receptors are different on each cell, and therefore, with a population of cells, a wide variety of antigens on a pathogen can be recognized. So unlike the innate pattern recognition receptors, which are dealing with motifs shared by lots of different pathogens, the adaptive immune system has things like the antibodies, the B cell receptor, the T cell receptor, which will recognize specific unique structures present on just about any organism that they happen to be able to encounter. There are two main types of cells responsible for adaptive immunity: B cells to produce antibody, and T cells to either coordinate the immune response, and we call those helper T cells, or to kill virally infected or otherwise infected, intracellular infected cells, and we call those cytotoxic T cells. Now, the whole adaptive immune system revolves around the clonal selection theory. And this underpins a lot of our understanding of immune responsiveness. First postulated by Burnett and subsequently clearly a central aspect of immunobiology. Basically, the theory goes like this, that in a population of lymphocytes, each one will have a slightly distinct receptor generated during that formation of each individual lymphocyte. So when an antigen or a pathogen bearing an antigen comes into the picture, one of those receptors is likely to be able to bind one or more motifs on this pathogen. That lymphocyte will then be selected to expand to create a clone army, if you like, of effector cells, each of which is directed, having a receptor which is directed at that initial antigenic motif. Following, so that's enough to clear the infection most of the time. Following that, some of these cells persist as memory cells, which will then respond much faster should that pathogen try the same trick again. Now, on the other flip side of this theory is that we must have a method of self-tolerance, but because our army of lymphocytes can recognize such a large variety of structures, it's likely that those structures are already present in self. So we don't want to create a situation where these cells are attacking our own self tissues, so we have to have a method of tolerance. So a lot of work has been done, and I will summarize that in this series of lectures, pointing out how tolerance is achieved. Okay, so now I want you to, I want to show you this YouTube video which was produced to commemorate the 50th anniversary of Burnett's theory. It's at that address. Have a look at it. I will show it to you now before closing part 1.
In 1960, Sir Frank Macfarlane Burnet was awarded the Nobel Prize for his contribution to the field of immunology. Burnet's clonal selection theory revolutionized our understanding of the immune system. To this day, Burnet's research remains the foundation of the way we think about disease and how the body responds to it. Clonal selection is the central mechanism by which your body fights infection. The following visualization presents clonal selection during an infection by strep throat bacteria. This patient has a strep bacterial infection causing a red, sore, pussy throat. The tiny round strep bacteria thrive on throat tissue. The bacteria are coated with hairs called M protein. M protein is what your body recognizes to be foreign. In response, your body will launch an immune attack against it. Your lymphatic network is an important part of the immune system, detecting any infections that arise in your body. The M protein hairs drain into the lymphatic system around this patient's neck. The lymph node is crammed full of B cells. B cells are guardians of your immune system, scanning the body for foreign material. Arriving at the lymph node are M protein hairs from the bacteria. Your body contains billions of B cells. Each B cell is unique and is sensitive to only one kind of foreign infection. By chance, one B cell is sensitive to the M protein hairs from the bacteria. This is the molecular surface of that B cell, magnified 10 million times. Its surface is covered with receptors that recognize M protein. The receptors grab onto the M protein hairs. This triggers an activation signal to travel into the cell. The activated B cell migrates and begins to replicate. In a few days, thousands of copies of the selected cell are made. This is the central idea in Burnett's clonal selection theory, where one cell is selected out of billions of B cells and identical clones have been created from it. Therefore, the cell is clonally selected. This creates an army of B cells that mass produce M protein antibodies and release them into the surrounding fluid. The antibodies circulate in the blood and travel into tissues. They bind to the M protein hairs on the strep bacteria. This flags them for destruction by the immune system. Other immune cells ingest the antibody-covered bacteria. They destroy the bacteria and eliminate the infection.
Well, I love that animation. I hope that makes sense to you. That's the end of part one. We've got a couple of parts to follow, so don't tune out yet. Before we do, I just wanted to draw your attention to the learning activities which I run in clinical immunobiology. So if you want to know more about clinical immunobiology, I have written a book which covers this course called the Primer of Clinical Immunology. It's available on Kindle, Apple Books, and Amazon.com as a print version. It covers what we cover in the course in its entirety. This video, of course, is, as I mentioned, chapter 0. So they are all the links if you wish to quickly scan them. I also have, of course, this video course in its entirety. So this is chapter 0. The chapters 1 through 25 you can find over on the Vimeo site, either as Immunology for Physicians, Immunology for BPTs. So there you will find the course in entirety, for which this lecture is the introduction. I also have a book and streamed video lectures on MCQs for those who are preparing for a barrier exam. That's the ebook, and again it's available in the same sites as I mentioned earlier to go with the video stream of the MCQs and the worked answers. I also have a Patreon teaching site. This is like an online community of people interested in learning about immunology. Its address is there. Basically, every week or so I post new learning exercises, which include revision exercises, mini lectures, MCQs, various news and updates every so often. So there's now over 2 years of accumulated material over on the Patreon site. So head across and check it out. You can get access to all those materials for a small monthly fee, all of which I donate to the World Wildlife Foundation. Finally, I run an Instagram site called Immunobiology Question of the Day, where every few days I post a new question on immunobiology along with a quick answer. They're quick, short, sharp revision exercises just to remind you of things that we have covered in the book and in the course. So check out all those learning activities. All the direct links are over on immunologyforphysicians.com. Have a bit of a break and we'll get stuck into the next part of this lecture.